Welcome to The Explainer. Have you ever thought about what it takes to be a zookeeper for the invisible? If you run a regular zoo, you know exactly what the lions and the elephants need to eat, how much space they need, what temperatures keep them healthy. But what happens when your entire menagerie is completely invisible to the naked eye? Today, we are diving into the fascinating manual scientists use to keep microorganisms alive, happy, and multiplying in the laboratory. All right, let's open up this microbial caretaker's manual. We've got 5 massive challenges to tackle today: feeding the invisible world, creating recipes for microbial growth, catching a single microbe, surviving without oxygen, and finally, freezing microbes in time. Challenge number 1: feeding the invisible world, the building blocks of microbial life. Seriously, what do you feed something you can't even see? Just like us, every single microbe needs a highly specific cocktail of elements just to keep the lights on. They need these essential macronutrients in large amounts to build cellular structures and generate energy. Carbon is the absolute non-negotiable backbone for all organic molecules. Then you've got nitrogen for proteins, hydrogen and oxygen, phosphorus for ATP, which is basically cellular energy, and sulfur. Without this precise elemental building block mix, microbial life literally just hits a brick wall. So how do they actually get these nutrients? Well, microbes are categorized by their diet, and most human pathogens, including familiar faces like E. coli, fall into a category called chemoheterotrophs. Honestly, they're remarkably similar to us in their dietary needs. They rely entirely on organic compounds to supply both their energy and that vital carbon backbone. But if you zoom out a bit, the diversity of microbial diets is just mind-blowing. You've got photoautotrophs that literally eat sunlight and carbon dioxide. Then there are the incredibly complex chemical diets of the lithotrophs, right alongside those chemoheterotrophs we just talked about. It really goes to show that to be a successful microbial zookeeper, you absolutely have to match the meal to the microbe. Moving right along to Challenge 2, recipes for microbial growth. Housing the invisible? Let's look at how we actually create these specialized environments in the lab. Think of this as the ultimate microbial recipe. You start with distilled water as your master solvent, mix in some peptone for your basic carbon and nitrogen, and toss in beef or yeast extract for those crucial vitamins and minerals. But the absolute star ingredient? Agar-agar. This stuff is magical. It's extracted from red algae and it melts at exactly 100 degrees Celsius. But get this, it won't solidify again until it cools all the way down to 45 degrees. That gives scientists the perfect window to mix in heat-sensitive nutrients before it hardens. Plus, most microbes can't even digest it, meaning they literally can't eat the floor right out from under them. And just by tweaking that agar concentration, we completely change the habitat. Bump it up to about 1.5 or 2%, and you get a firm, solid surface, perfect for trapping bacteria in place so they form visible, isolated colonies. But dial it back to just 0.2 to 0.5%, now you've got a loose, semi-solid jelly. This is brilliant because it's thin enough to let us actually watch motile bacteria swim right through the medium, while the non-motile ones just stay stubbornly stuck in place. We can customize these recipes even further depending on our end goal. Got an incredibly picky eater? We create enriched media by adding blood or serum. Need to weed out the riffraff? We use selective media, loading it up with antibiotics to inhibit the microbes we don't want. Or we can use differential media, which actually changes color to instantly identify bacteria based on specific biological traits, like whether or not they ferment lactose. Which brings us to Challenge 3, catching a single microbe, the art of isolation. You really can't study a chaotic mix of hundreds of species at once. You need pure isolation. But how do you separate just one microbe from a herd of billions? The solution is a remarkably elegant technique called the streak plate method. We take a mixed sample on a sterile metal loop and mechanically dilute it by wiping it back and forth across the agar. The absolute key here is sterilizing, or flaming, the loop between each section. With every pass, we're dragging fewer and fewer cells until individual microbes drop off, completely separated from the pack. It is so satisfying to see this in action. The heavy, completely blended growth at the very start gets progressively thinned out. You sterilize the loop, drag some bacteria into the next zone, sterilize again, drag again, until finally the cells are so diluted that they grow into beautiful, distinct dots. Every single one of those well-isolated colonies is a pure culture, grown from just one single parent cell. Now for Challenge 4, surviving without oxygen, cultivating anaerobes. So far, we've talked about microbes that love the open air, but what about the ones that don't? This is a serious paradox for our lab caretakers. If atmospheric oxygen is literally toxic to the microbe you're trying to study, how on earth do you safely house it in a lab entirely surrounded by breathable air? One incredibly clever workaround involves using liquid broths packed with reducing agents, like thioglycolate. These chemicals aggressively react with dissolved oxygen and reduce it to water. Even if the top of the tube is totally exposed to the air, the thioglycolate strips the oxygen out as you go deeper. That allows those hypersensitive anaerobes to hide out and thrive at the very bottom, completely shielded from the toxic oxygen above. But what if you need those solid agroplates we talked about earlier? For that, we engineer a completely anoxic environment using an airtight anaerobic jar. A chemical packet inside generates hydrogen and carbon dioxide gas, but the real magic happens with the palladium catalyst pellets. They force that newly generated hydrogen to react with any rogue oxygen left in the jar, turning it harmlessly into condensation. It essentially purges the entire atmosphere of oxygen. And finally, Challenge 5, freezing microbes in time, maintenance and preservation. Once you've successfully fed, housed, and isolated your pure culture, you have to preserve it. We essentially have two modes here. For short-term preservation, simple refrigeration at 0 to 4 degrees Celsius works just fine. It slows down the microbe's metabolism enough to keep it stable for a few weeks, though you still have to subculture it, moving it to fresh food before it starves. Long-term methods, though, that's a whole different ballgame. Techniques like paraffin oil overlays, cryopreservation, or lyophilization are designed to completely halt cellular activity for years without killing the cells. Take cryopreservation. We are talking about subjecting living cultures to a shocking negative 196 degrees Celsius in liquid nitrogen. But there's a massive catch. When water inside a cell freezes rapidly, it forms these jagged ice crystals that can literally slice the cell membrane apart from the inside. To prevent that, we have to mix in stabilizing agents like glycerol. It acts exactly like microscopic antifreeze, letting the cell freeze solid without bursting. But if we're talking about the absolute gold standard, the method used by the world's premier culture collections, it's lyophilization, freeze drying. The microbe gets rapidly frozen at negative 70 degrees Celsius, and then it's subjected to a powerful vacuum. This causes all the water inside the cell to sublimate. It flashes directly from solid ice into a gas, completely bypassing the liquid phase. What's left behind is a dry, dormant powder of perfectly preserved cells. The results of that freeze-drying process are just staggering. Those cells remain totally viable, ready to be woken up with a little water and warmth for over 20 years. As we wrap up this explainer, it truly leaves us wondering, if we can safely hit pause on biological life for over 2 decades just by manipulating their environments, what other incredible survival mechanisms are hiding out there in the microscopic world, just waiting to be unlocked?